take no more chances then and there. I got it started.
I have a question, Charlie.
Yes, thank you.
Are we gonna have practice quizzes before these tests?
Yes, you are.
Okay, great.
I won't give them to you until Friday and Monday, or over the weekend. Okay. I do have two already to go, but you haven't, in my opinion, uh, had enough experience with the material yet. And if you were to run into a practice quiz that's got questions that you haven't had yet, or you don't have a full understanding of, that'll be discouraging. So I'd rather wait just a little longer until I think that you really have a chance of answering every one of those questions.
Okay, thank you.
Okay. I sent you the second email, had an attachment. Uh, I've already actually put it on the module as well. And that's the second bonus exercise worth 4 points. Odd number 4, but 4 points for that, 7 points for uh, all of the conference zooms if they're made, and the other 4 points is for the histology study guide we went over yesterday. That may again seem like an odd number, but along with the points you've had previously, it totals 40. Okay, and that's what I promised you. So the instructions for that mm, histology, uh, what do they call it? I forgot what the title was. Uh, but the bonus exercise, that's what it was called. Uh, the instructions are very simple. I sent it to you because I thought it'd be easier for you if you have a computer or a laptop or a tablet to download it there, open it up, and it's just a Word document. It's not protected. You can go ahead and write your answers right there. And you can save it as you go along because you probably don't know all the answers right now. But you can get a start on it. You could definitely get a start on it, plus a preview. I really want to encourage you to start. Okay, don't wait to the last minute on any of this. One of the ways I did that is I need to uh, be busy on the last day too, and so these two bonus exercises worth 4 points apiece are due Wednesday, and they will not be taken any later. Okay, just absolutely not going to be taken with enough time everybody's had to get started on it. Now, I don't know if the, uh, what I observed amounts to anything or not, but I went in and looked at you, uh, the class, everybody individually, and saw when the last time you were looking at our material was. Okay. And only two people were looking at anything last night. Well, maybe you're not you got the pace. Maybe you don't have the wrath or fear of God yet, but only two people looking at it, and 32 getting behind, possibly. Okay, maybe you did it afterwards. I usually go to sleep around 4 o'clock, but I went to bed early last night. I went at 1 o'clock, and I could have missed somebody's presence. When I look at things like that, I just want to get the feeling for, is the class responding? Are they working towards the grades that they want? Okay? I'm not doing it for any other reasons. There's no points involved or anything like that. Okay. And to be honest with you, I don't remember who was there the longest. I don't remember even who the two were. It doesn't matter. Okay. I did look also at the videos to see if they're being looked at. And of course, last night's video wasn't looked at yet. It got there late. But the previous one was looked at by 28 people. and the other one before that was looked at 41 times. That means people looking at it more than once. So the videos are being used, and if that helps, well, there it is. You got the tool. Okay. What else? I think today is probably going to be the last of the, if I call it that, leisure days. Okay. Uh, we have a lot of material to cover, but I don't think it's what you'd call intense. Thursday and Wednesday, Wednesday and Thursday are going to be a little bit more intense. Okay. The material just happens to be that way. If you have started your reading, great. I have no idea about that. But you asked me, or somebody, probably was Mike, I bet you, uh, am I going to outline what needs to be read and doesn't need to be read? Okay. I said no because I've assigned the entire chapter. However, you know that you don't outline applications to health. You don't outline clinical observations or what looks like reference material. Okay. I am not going to be grading these on what you did and decided not to outline. That's not going to happen. Instead, I'm expecting that you wanting a good grade will outline those portions that follow along with our lecture material, okay, and seem important to you, okay. I think at this stage of outlining a few chapters, and you did not have as many chapters given to you as a normal semester. It's just too hard in the summer. But I think you have a grasp about what needs to be outlined or not outlined. Okay. And as I said, I will accept the outline uh, uh, in your in the condition you give it to me, assuming and believing that you gave an honest, good attempt at it. Okay. I don't know if that came out right, but okay. Okay, let's get into it. Now, I tried a moment ago. I came in early and I asked, can you see the cursor? Can you?
Yes.
Yes.
Right. You can see the cursor. Okay. But we're still not where we want to be. So I still have to apparently log in. That's not going to work. But it's a bit faster. Okay. Yeah. If you haven't looked at it this morning, you'll see that module 7 has enlarged from here on down. The assignment is not worth any points. Uh, the assignment is the 2 outlines. Those are necessary next Wednesday also, or before, in order to accumulate any of the Zoom points. You know that. The discussion, I'll leave it up to you. It's not going to affect your grade one way or another if you want to participate in the discussion. A few people definitely want to. They have their feelings about these, uh, about stem cell. Okay. I certainly do too. So I'll put my two cents in there and you can add to it, or maybe you'll get to it first. We did the integumentary system at a starting pace anyway yesterday. The histology study guide we looked over. That's the one where you put definitions or functions, and that's due for 4 points on next Wednesday. Now, if doing it seems uh, like it's taking a lot of time, you may want to back out and not take the 4 points, but that would be a mistake. First, you want the 4 points, okay? And second, this stuff in the study guide, it's almost word for word. Every word in it's going to be on one of the three tests that we have. Very important little piece of paper. We looked at the tissues to begin with, part one and part two. And then we finished off the recording. And now I'm going to do histology photos in a moment. But first, I'm going to start with lecture. And we're starting the nervous tissue. And if you notice, we have nervous tissue today. We have the autonomic nervous system tomorrow. And we have spinal nerves and the autonomic nervous system on Thursday. So this is a 3-day event. Okay. It starts off easy. Thank God. That's what I meant by a leisure day. Okay. The nervous system has three main functions. One is to conduct stimuli towards the central nervous system, which is the brain and the spinal cord. The other is to conduct motor movement impulses away from the central nervous system out towards all of the body organs. So the central nervous system has to receive a stimulus and then it goes out. You can get some motion without making this complete circuit. It's called a reflex arc. If you were to touch a hot stove, say an electric stove, and it's been turned off just recently, you know, one of the burners is hot. Which one? Well, if you're as stupid as I am, you'll take your hand and put it flat on a burner until you find the one that just makes you scream because it's so hot. Okay, well, when you hit that one, it's hot and your hand pulls back immediately. The sensation, the sensory stimuli, is going towards the central nervous system. Okay, it doesn't reach the brain yet. It reaches the spinal cord. It reaches the horns of the gray matter. Now, maybe you don't remember that from a week or so ago, but we'll be reviewing it, don't worry. It reaches the spinal cord and then immediately cuts across and goes back and tells you to pull your arm away. Now, that painful uh, scenario of heat coming in and then the hand pulling away involves incoming and outgoing impulses, a stimulus coming in and a motor impulse going out. What about the brain? Well, you know as well as I do. You touch a hot stove like that, your hand moves even before your brain has access to that information. But your brain's going to be told in a split second, literally. And so it does travel onward to the brain. No reaction, no nervous reaction takes place without the stimulus reaching the central nervous system. Okay. An exception to that, somebody could say, well, what about an eye that twitches on its own and you can't stop it? Okay. What about an arm where a vein or an artery inside of it is pulsing every once in a while? I have no answer for that one. I don't. Okay, but a normal sequence is the stimulus comes in and the motor impulse goes out. Sometimes the stimulus can come in and the brain can get it and think, so what? I don't care. Don't do anything about it. Okay. No, you got your answer there, and you'll have incoming but no outgoing. The third thing that the nervous system does is to maintain homeostasis. We defined that before as the maintenance of the body as is when it's in a healthy condition. Okay. So the nervous system is basically responsible for these three things. The components, first off, the central nervous system itself. That's the brain and the spinal cord, and that's all. The brain and spinal cord from chapter, I believe it was back at 1. The brain and the spinal cord are located within what's called the dorsal body cavity. Okay. If you could take the brain and spinal cord magically out of a human being, the cavity long at the bottom and rounded at the top is the dorsal body cavity. Okay, well, the central nervous system can send out information as motor impulses and can receive information, but on its own can't do anything. It has to be able to innervate something, make something work. I don't know if you ever saw the stupid movie, it was a science fiction movie where brains chase people around. It was the dumbest movie I ever remember. The brains didn't have feet or anything, but they were able to move. Okay, central nervous system is one part. The second part, just dividing it in half, is the peripheral nervous system, PNS. We always use these abbreviations. The peripheral nervous system is located outside the central nervous system. It receives information as well, and it allows motor neurons or motor impulses to travel its routes. We have in routes, out routes. Well, what are we talking about? The afferent nervous system. Sensory receptors travel towards the central nervous system. Okay. Sensory neurons, I have written here, travel towards the CNS. Remember, afferent means an affinity for something, or a love for something, or an attraction for something. And the neurons in the afferent nervous system are sending their sensory information to the central nervous system. Going the other way, we have motor neurons leaving the CNS. Okay. And these are the ones that would, for instance, go to skeletal muscle, much smooth muscle, but not all of it, mostly skeletal muscle. We also have the autonomic nervous system. We won't get into that today. We will save that for tomorrow. The autonomic nervous system tends to scare people. It's actually a delight. Okay? But the autonomic nervous system innervates smooth and cardiac muscle and glandular epithelium. The term innervates important. It doesn't make a lot of sense to people right off the bat. It means to have an effect. The autonomic nervous system has an effect upon smooth muscle and cardiac muscle and glandular epithelium. If I were to use a human example, you walk over to a light switch and turn it on. You just innervated the light switch. Okay. You put the keys in the car, you start it up, you just innervated the car, you had an effect upon it. So it's not a complicated word. The autonomic nervous system is broken into two subparts, the sympathetic and the parasympathetic nervous systems. Again, we will reserve that for tomorrow. And then we have the cranial nerves. The cranial nerves, there are 12 cranial nerves. You'll be asked their function, and you'll be asked to name them. And they'll be given to you by Roman numerals 1 through 12, and you'll be asked to name them and just state briefly their function. I have prepared a chart for you. I'll show it to you later today. It's at the very bottom of Unit 7 here. And I figure you can easily learn that in half an hour, probably half an hour. I have this in a chart form. Okay, you can read the reading, it's just repeat. Here's the brain and spinal cord up here in this rectangle. This is the central nervous system then. Everything else outside of the central nervous system is located in the dorsal body cavity. So all of these boxes, without even reading them yet, are part of the peripheral nervous system. Peripheral. Okay, well, we have information from the peripheral system that serves as incoming sensory information. Okay. This takes information in to both the spinal cord and the brain. The afferent fibers. Fiber is another term that is used for neuron. Fiber is also used to indicate a pathway. How many neurons do you think have to be involved in transmitting the pain of an elephant stepping on a thorn to the elephant's brain? How many neurons would you imagine?
Maybe
This is
just
where we
one.
take a guess.
100?
Okay, that's a good guess. Thank you. It's wrong, but you knew it would be.
I'm going to guess thousands.
1,000. Okay. Anybody want to bid higher?
Million?
Okay.
3 million?
Okay. I wish I could take that bet. The answer is 2. There are just 2 neurons. In most cases, there are exceptions, but 2. The one that receives the stimulus of pain from stepping on the thorn transports it, depending upon which system it follows, transports it almost up to the spine of the elephant. It synapses, that means joins with the next neuron, which takes it all the way to the brain. Okay, just 2. Now that's the same for you too. We don't have a lot of neurons to make a chain. Why not? Because every time we have the end of one neuron, we have to change neurons. It's like changing trains. It's like changing planes at the airport. All this changing takes time. And the essence of speed is necessary to protect the organism. In other words, if you have a sensation coming in, good or bad, you want to know about it right now. So the fewer neurons, the better it is. Now, can you think of, just imagine, I know you haven't seen one yet, just imagine a neuron, a nervous neuron, okay? Be kind of flimsy, wouldn't it be on its own? If you only had one neuron and then the second one going from your toe to your brain, what are the chances in your lifetime that you won't break that neuron? You probably would. And then what would you do? Okay. And think of all the other neurons and what they do going the other direction. We have neurons in bundles. We don't just have a single neuron, we have a cluster. It's not that we have a single strand of spaghetti uncooked, but we have a handful to make a real fiber. Okay. By having lots and lots of neurons run parallel to each other and do the same thing redundantly, then we're protected. We can break some. The others take over. The others continue with uh, their task. Okay, so we don't want many neurons in a pathway, but we want a lot of neurons in the pathway. Same going the other way. Incoming is the afferent system, afferent nervous system. Here it's being called the afferent fibers. Leaving the spinal cord to lead through the ventral root. I know you don't remember that. Some of you do. But the ventral root goes out, and then you reach the efferent fibers. These are the outgoing fibers that carry the information to what? Over here, skeletal muscle, and over here in the autonomic nervous system, smooth muscle, cardiac muscle, and the glandular epithelium. The outgoing, the incoming, you only need one type of incoming. You got pain, you got pressure, whatever, you got a neuron for that. But what's the reaction going to be? It can be several things. It can first off be, hey, trouble's coming. Let's get the hell out of here. Legs start moving. Okay, so you've got skeletal muscle that can be innervated. This is by the somatic nervous system. Somatic meaning the body. Okay. This is skeletal muscle. This is the only place it resides, right here. Well, we have the autonomic nervous system, and again, we won't get deep into it today, but just a little here. The autonomic nervous system takes information from the CNS, again, to smooth muscle, cardiac muscle, and all glands. Glands are composed, simply enough, of glandular epithelium. I didn't show you that one yesterday, but it's the easiest one to learn. The autonomic nervous system, we'll find tomorrow, is broken into two parts. The sympathetic portion, also known as thoracolumbar, the sympathetic nervous system, and the parasympathetic nervous system, also known as the craniosacral. That thoracolumbar and craniosacral will be explained to you tomorrow. Okay. Basically, the sympathetic nervous system acts when you're in danger. It initiates fight or flight responses in all of your internal organs, that's smooth muscle, your heart, that's cardiac muscle, and many glands, depending upon what the danger is. The rest of the time, when things are calmer, the parasympathetic system will predominate. Okay, you can keep things more on an even keel. These two are not exactly opposite each other, but close enough that there's no question they're going to be designed to fool you on that. Okay, this chart will appear again in a different form for you. When we look at the brain and the spinal cord, we think of, oh, yeah, that's all nervous tissue up in there. Actually, it all is called nervous tissue, but 90% of the cells in the brain are not neurons. They are called glial cells. 90% of the cells in your brain and spinal cord do not conduct anything. They help support all the 10% that do. And these are the glial cells. They're not functional for passing along information and initiating a motor response. When somebody tells you you lose 1,000 brain cells a day, they're probably right. When somebody adds to that, oh, if you drink a beer, you lose another 1,000. And if you get a hangover on top of everything else, you lose another 10,000. Well, they're probably right. I've seen some studies on that, and I don't see any reason to disagree with it. But you have a lot of brain cells, like 4 billion to 6 billion brain cells. But unfortunately, 90% of them don't do any thinking. So you got to protect the other 10%. Okay, 90% are glial cells. So let's see what those are. First are the astrocytes. The astrocyte looks sort of like this one. Extensions in all directions, a nucleus in the center, and it functions to support the rest of the neurons and blood vessels and other astrocytes and glial cells. It's the glue that holds everything together. Here's an astrocyte here in this mock-up. Here's an astrocyte. Here's a neuron. And I'm going to call it blue. Let me get away with it if I'm wrong. Here's another neuron. And this astrocyte has these foot-like extensions that will bind the neurons to each other, or even to this, which looks like a small capillary. Yes, it is. So things hold together. That's the function of the astrocyte, and there's just billions of them. The oligodendrocyte. Okay. The oligodendrocyte is shown here, I believe, in blue also. Okay. In gold, in the middle, is a neuron. We haven't talked about the parts of the neuron yet, but we're just about to. But when a neuron fires an impulse, it goes in this direction. And then it goes to here, where it may cross over and touch a second neuron. Remember, there can be there's 2, sometimes there's 3 or even 4, but usually 2. Okay, so it can branch. But if we need a neuron to fire fast, certain responses have to be done faster than others. These oligodendrocytes in blue have extensions that coat the axon running through them. This one coats here. Now there are no gaps. There would be coating here as well by another oligodendrocyte probably. So if the whole axon is coated, and that coating is called myelination. Myelination. I'll find you here it is. the myelin sheath. Okay. It allows electrical messages to travel faster. It's also what gives white matter. We talked about that once before with the cervical vertebra. It gives white matter its name. The myelination always is a fatty substance and it appears white. These oligodendrocytes can also support because they can interconnect with their other extensions to capillaries, other neurons, other oligodendrocytes, other glial cells. This guy here is another astrocyte. It changed the color, okay, but it's a glial cell. So an oligodendrocyte, it's a glial cell. Here's another astrocyte over here. I'll come back to the neuron itself in a moment. There's a third type of glial cell called the microglia. It lives up to its name. It's the smallest. 15% at most, 10 to 15, of all the cells in the CNS are microglial cells. And they are the immunological defense for the brain and the spinal cord. They defeat foreign agents, bacteria that can sometimes get in. Cellular debris will not accumulate. Red blood cells will die any place in the body at any time, although the spleen and the liver are the greatest repositories for red blood cells that are broken up. If they break up in the brain, the oligodendrocytes will phagocytose them and eat them up. The microglia, that's a small one here, is capable of diapedesis. We used that term, I think, twice before. That means being able to flatten out or change its shape to squeeze in between cells. The microglia, although it does have extensions as you see here, never forms permanent attachments because it needs to move around. So its attachments can be released. Here's the oligodendrocyte, extensions forming part of the myelin sheath. Here's the astrocyte. And over here is a neuron. Okay. We'll get to the neuron as I promised. Types of neurons. Okay. As I mentioned a moment ago, we don't just have one neuron running a course, taking a stimulus or a motor impulse. We have a group of neurons, thousands possibly, neurons running side by side, more or less like a cord. Okay. And these neurons can be traveling towards the central nervous system or away. But when they are bundled, when you have a bundle of neurons, it's a nerve. Okay. We have afferent nerves and efferent nerves. We also have nerves that run in both directions. Okay, so you're on a road in a car, you're driving towards some monument in the center of the city. You're on an afferent roadway. It's one way going into the central nervous system or the monument that I predicted here, okay? Or you can come away from the monument on a one-way road away, and that's an efferent pathway. But you and I both know that most of the roads around here are two-way. We have traffic running both ways. And that's a mixed nerve. Okay. We have sensory nerves, which are also known as afferent nerves. We have motor nerves, which are efferent nerves, and most of the nerves are actually two-way streets. Okay, so within the nerve, you have fibers running in and out. These are mixed nerves. Mixed nerves are the most abundant, most numerous. Here are three types of neurons. Let's try it a little different this time. I'm going to go through this reading first and then the diagram. It may be better to do it the other way, but I just felt like I wanted to try it. Okay, the first type of neuron is called a multipolar neuron. It is the most common. Oh, I'm famous, you know that, for asking what's the most common this or that. The multineuron is the most common. Stimuli reach through its receptor arms or dendrites. Okay, the way into the multipolar neuron is through what we call a dendrite. There can be several of these dendrites. These are all dendrites. This is a dendrite. I guess I can't do it without referring to a diagram. So the sensations or stimuli come into the dendrites first. And then that causes what we call depolarization. Now, fortunately for you and for me, to try to explain it, depolarization is the property of the physiology class. They don't want me to explain it wrong or in a way that they wouldn't do it. So we'll basically say that for them. But I can tell you this. When a stimulus reaches the dendrite, it causes what we call a depolarization. Before the stimulus reaches the dendrite, there is more sodium, that's a cation, Na plus, on the outside of the neuron. When the stimulus reaches the dendrite, it causes the release of calcium and an influx of the sodium. Before the stimulus reached the uh, dendrite was polarized, more sodium outside, more positive charge outside. Once the stimulus reaches it, it flushes and you get the sodium to rush in. And that's called depolarization. That generates a signal that affects the next cell, the next cell, the next cell. It's sort of like the chain of events where one domino topples and causes the next one to move and the next one to move. All of this motion or depolarization ends up accumulating in a portion called the axon hillock. This is the axon hillock right here, just before the axon itself leaves the cell body. Okay, I'm going to move to a picture now for the rest of it. My little experiment, I think, failed. I didn't like it. I think you're okay though. I'm going to do it again if I need to. Okay, can't get this one any larger. Here's the cell body of a multipolar neuron. Here, these tree-like or feather-like extensions These are the dendrites. Every dendrite that is depolarized sends a signal towards the axon hillock. Maybe only one of these dendrites gets a signal. It's not going to be enough to activate anything. Maybe two of them, maybe all of them get the signal, and that's going to be enough to activate the axon hillock. If the axon hillock is sufficiently depolarized, this part here, then an impulse is generated that goes down the axon cylinder, also known simply as the axon. They have it here as axon. That's fine. If this axon is myelinated, and it is here, see myelin sheath, then it will transfer the impulse rapidly. It goes to the other end, and then it can flare out and go to different neurons. These are called axon branches, axon terminals, or the proper name is teleodendrion. The influence coming from the axon cylinder can be divided up and sent to several neurons so that they can sort of snowball the effect in going the rest of the distance. A synapse, we'll talk about that separately, is where one neuron meets the next neuron. Now, you may notice in this one picture the term Schwann cell. Well, this is the myelin sheath. And a moment ago, I showed you the oligodendrocyte making up the myelin sheath. But there's another type of cell called the Schwann cell that also myelinates. The oligodendrocyte that we saw earlier here myelinates axons that are in the central nervous system. In the peripheral nervous system, a cell of different origin called the Schwann cells do the myelination. The gap between myelinated portions, these little teeny gaps, I'm sorry this won't enlarge, these are called the nodes of Ranvier. Now I know somebody's told me that's a French word. I don't speak French, so I can't help out if I got it wrong. You can correct me and we'll get it right today, but I'll forget it by the time we come back to it. 2% of the world's population are native French speakers. Why did we all go to take French class? I don't know. Maybe we don't do it anymore. In my day, we used to take Latin class. What a waste that was. Okay. Let me quickly review the multipolar neuron and why they call it that. It's multipolar neuron because there's multiple ways in. I'm touching different dendrites, and multiple ways out. It's multipolar. The bipolar neuron is much easier to look at and to understand. Bipolar has one way in and one way out. Well, if there's only one way in and one way out, why even bother to go in? Just keep on going on through it. Okay? Don't know the answer to that myself. However, whatever is picked up by these dendrite trees that you see here, this is the dendrite. They all coalesce into one structure, which is honestly an axon. But we're going to call it a dendrite because the books do. The information comes in, passes through the cell body, and then keeps on going along the axon to the axon terminals or teleodendria. The previous neuron, the multipolar, can take in information from many sides and then deliver it to many locations. The bipolar can get one piece of information and take it and send it along without changing it. Bipolar is found in the eye, where red is red, blue is blue, and yellow is yellow, and they don't mix. And in the ear, where sounds at different pitches are only stimulated by that pitch, and then it doesn't change. It's sent through the vestibulocochlear nerve to the brain. So one way in, one way out. Then we have the third type. This is the pseudo-unipolar neuron. It's actually called in older books the unipolar neuron. In fact, I learned it that way too, but they've changed the name to the one I have in bold here, the pseudo-unipolar neuron. They have a cell body with two extensions, and both are technically axons, one extending to the periphery and the other towards the spinal cord. Okay, these are sensory neurons of the peripheral nervous system, and these are the ones that we think of the most in the arm: pain, pressure, heat, cold, movement of an insect or an ant or anything. The wind, we can feel it on our arm. Okay, so these are pretty basic stimuli, and they move along from the periphery by the pseudo-unipolar neuron, which is here. Here's the cell body. Here's a dendrite, another dendrite, another dendrite. But they all come together into one axon. That axon takes a turn off for a moment, stopping by the cell body, and then continues on and can change. It can only bring in one type of stimulus. Why? Because they all have to accumulate here in this single tract of the axon. These have their cell body off to the side. Notice that the bipolar, which looks similar, has the cell body in the center. Think in your mind, and you're going to take a piece of rope. Okay, maybe 2 feet of rope. Tie a simple knot in the center. Okay, set it on the table. Get another piece of rope, same size, make another knot, set it in the center, but put the knots on top of each other or next to each other. Keep on doing this, keep on doing this. And pretty soon, you're going to get quite a few strands going in either direction. But in the center, it doesn't show here, you get a cluster of these cell bodies. And it makes a bump. And that was the dorsal root ganglion that we saw in that vertebra last week, the dorsal root ganglion. It's all the cell bodies of these pseudo-unipolar neurons. This is coming from the skin. This is passing through right outside the spinal nerve. And then this is going the rest of the way on the dorsal root to get into the spinal cord. Initial body is a term you do not need. Some vocabulary. We've used most of these terms already. That's good. But you need to be at least mildly or fully familiar with some of these. For instance, a nerve. Okay. We've talked about that, so I'm not going to do it again. A nerve fiber. Okay. It's really the same thing, but just to get the idea that there's a lot of it. Afferent fibers incoming, efferent fibers outgoing, somatic to the body, visceral to the organs. A neuron, it's not only just a nerve cell consisting of a cell body and its processes, the neuron is also the physiological unit of the nervous system. The axon, usually it's a single straight fiber that conducts an impulse. Now, sometimes it can be double, but let's forget the sometime. Dendrites, okay, these receive the stimulus. The term perikaryon keeps showing up, and the perikaryon here is the cell body. Cell body is also known as perikaryon. Peripheral process in a bipolar neuron, which this one is bipolar. The peripheral process is out to the body coming in. The central process is coming in, but it's past the cell body. A receptor, anything that can detect a change in the environment. A receptor is a receptor, stimulus receptor. Effector, this is a tricky term. An effector is usually a muscle or a gland. It's something that is effected. Now, to me, I know the first time I saw it, I thought effector must be a noun. That must be the person or thing that's doing the making the effect. It's not true. The effector is a muscle or the gland that is being affected. The functional contact between two neurons are called synapses, and we'll be going into them shortly. This is a cell body, a perikaryon. An axon, very simplified drawing, obviously. Axon branches. They would then meet dendrites not drawn in, a perikaryon, an axon branch. And then this does not have to be a third neuron. This could now be the organ itself. It could be the muscle. It could be the stomach or anything else. Gray matter. Gray matter is a part of the nervous system in the brain. The horns that we looked at are gray matter. The gray matter is not myelinated. That's why it remains gray. Since it's not myelinated, impulses move through it comparatively more slowly. White matter has the aggregations of the myelinated axons. White matter is myelinated. White matter moves fast. If there are a cluster of nerve bodies, okay, and it's in the central nervous system, it's a nucleus. Another term or another way of using the term nucleus. Let's come right back to it. Let's move on to ganglion. We've all heard that one. A ganglion is a collection of nerve cell bodies outside the CNS. For instance, in the pseudo-unipolars that we just looked at, and I had you tie the knots and pile them together, that big clump of knots in the center would be a ganglion. It would be located in the peripheral nervous system. Uh, Even if it was part of the dorsal root ganglion, that's still out in the peripheral nervous system. Now, if these neural cell bodies were to accumulate inside of the brain, and they do, then they're called nuclei or a nucleus. Okay. I'd appreciate it if you have questions, bring them tomorrow. I'd appreciate it if you don't have questions, but you're lost a little bit. Bring that fact up, okay, so that we can go over it again. Also, if you begin chapter 12 on nervous tissue, I think it will help with the diagrams that they have. When we come back, we'll be looking at organ systems, the start of that, and what else we have yet to do for practicum number 4. Okay, so I'm giving you a break till 2:30. Okay? Okay.
Hey, Charlie.
Howdy. Okay, we'll just get moving because kind of. Okay, if you can see the cursor, if you can't say something. We just finished this brief talk on nervous tissue. This will be really reinforced if you begin that chapter. Okay. Look through the chapter, find the pictures of the different types of neurons, the multipolar, bipolar, and pseudo unipolar, and you'll be in the right place with that. We need to move down. We're actually up to histology photos. This is a chart. You have to open it here. Okay, this is the chart I showed you yesterday. And we went through these first two columns. Well, we've also gone through in the past skeletal muscle, cardiac muscle, and smooth muscle. So I don't want to go through those again. I can, but if you need me to, but just click on them and you'll see the three different types of muscle that we looked at once before. Okay. What I do want to just quickly show you is this neuron. Okay. And the neurons, of course, are the bigger parts. They're so big that when you put them under the microscope, invariably they break. Okay, but here's the best one, I think, right in here. This is the cell body, or the parakaryon. Here's a dendrite coming in. Here's a dendrite. Here's a dendrite. And here's a dendrite coming into this. Now, the cell body sort of terminates at a triangular structure right here. And that's the axon hillock. The axon hillock is where the depolarization ultimately accumulates. If there isn't enough depolarization, for instance, if only one or two of the dendrites receive impulses, that may not be enough to stimulate the axon hillock into action. And that action is to generate an impulse. And it doesn't happen until there's enough depolarization. Once there's enough depolarization coming in, passing through the cell body to the axon hillock fires, and an impulse propagated then through the axon cylinder, which ends here because it's broken. Okay, it's just broken right there, unfortunately, but expectedly. The axon hillock, the important thing is it's an all or none proposition. It either fires or it doesn't fire. If it doesn't get enough pull, it will not work. If it's like a trigger, if you're pulling a trigger and you don't pull it hard enough, nothing happens. If you pull it just exactly the least amount necessary to fire, it fires. It's all or nothing. Surrounding the multipolar neurons you see here, these are the glial cells, the nuclei of the glial cells. I'm going to be asking you to, on your own, go through and review these but I have not yet done these over here. And I'm going to do the final ones right now, okay, so that you can see the tissues at least for the first time. Salivary gland. I don't know if this is put what order this was put in. I put the chart together, but it was so many years ago. Here's the salivary gland. Oh, and I don't like that salivary gland. Okay. This is fat. We don't. We always expect to find that. Here's a duct right here, and this dark material. These are acinar cells. ACINAR. Acinar cells are those that produce enzymes, which are expelled from the cell, or actually from the organ, by ducts. The salivary gland is an exocrine gland. It expels its contents through a duct to a specific location. The tongue. We obviously don't have a specific order here, but you will be seeing this on the exam. When you see the tongue here, you see at the top, oh, that's got to be a stratified epithelium. And are the top flattened? No, they're not. So it's a transitional epithelium. But what you need to know about this structure are these bulbs here. And this slide gives it away with the TV. These are taste buds. I think I've shown you this slide before. That's okay. Anytime it is a repeat, it's a reinforcement, actually. This is the lining of the upper esophagus. Now, from your memory, maybe not today, but after you filled out that study chart, that histology study guide, most of these things do latch into your memory. You know the upper esophagus is lined by stratified squamous epithelium. This area right here with this bump, and this little bump, that's this here. So let's just look at the bigger picture. And when we look at the larger picture here, we see stratified and flat cells up at the top, very compressed cells up at the top. So this has got to be stratified squamous epithelium. Whether or not you can identify it as the esophagus, that would be problematic, and I would not ask you that. But I would ask you to look at this, see that it's multi-layered, that means stratified, and that the outer layer is flat, stratified squamous. The stomach, you would think that with as many pictures in the books that you are bound to find of the stomach, that it would be one of the easiest. It just takes a little bit of being calm to figure this guy out. First off, we obviously see these things here, but look at this area down here. This is smooth muscle. This is muscle. The lumen of this organ is way up here. The photograph, the way it was taken, doesn't show you much of the lumen. Okay. The lining of the stomach, we don't have to see this in clarity. The lining of the stomach is simple columnar epithelium, because the lining of all parts of the GI tract south of the upper part of the esophagus is lined by simple columnar epithelium. You may think for a second, and I can't blame you at all, that these look like villi. And if that were villi, it would have to be small intestine, the only organ that has villi. This is not villi. What this is chasms going down, not fingers going up, but splits. This is an artificial split here. Splits going downward to where the enzymatic pouches are, right down in here. Whether you're cutting down like this or you're raising things up like a seedling, you might get this appearance. But this really doesn't look at all like the villi. Look how smooth this is. That's stomach. The villi would be scattered. And I probably have a picture of that coming right up. Yes, I do. This is the simple columnar epithelium of the small intestine that I showed you yesterday. This is the same over here. Well, what the heck is it going in all different directions like that? Well, in the small intestine, we have the plicae circularis. If you recall those, the parts where it invaginates comes in as a small little growth or a ring going around, and they're lined with villi. This is a villus here to here. It's just not as pointed as this one. The large intestine gives itself away with the numerous goblet cells that contain oil. This lubricates the passing food. If you look at this picture, it always reminds me of some sort of community of octopi. Okay, because there you've got the suckers all showing. Function of the colon is reabsorption of water, compaction of waste. Always tell yourself, what is the function of what I'm looking at, so that you don't have to go back and do it again. This is pancreas. This is an islet of Langerhans here. It's light. Magnified, it's over here. This is an islet of Langerhans. And this down here is an islet of Langerhans. Why isn't it the same size? It probably is the same size. But if you have an apple floating in water and another apple and you push it part way down, that's what you're going to get. These are alpha and beta cells, which we'll talk about next week when we talk about the endocrine system. Okay. But surrounding the darker cells that you see here are the acinar cells of the pancreas. Remember, acinar means it secretes an enzyme. The enzyme being secreted by the pancreas is pancreatic amylase for the digestion of starch. Okay. The pancreatic amylase is produced by an exocrine gland because it has a duct, but the islets of Langerhans produce glucagon and insulin, which flows directly into the blood that passes through it. And this is blood here, here, and here, blood here, some blood here. This is liver. Okay. We've looked at the liver, and then looking at the models that I'm going to be showing you, you're going to see the liver again. In fact, you're going to see a lot of things for the second time, which shouldn't bother anybody. It looks like tiles. To me, it looks like tiles. And when you can see it in a model, they're usually six-sided tiles. In the center is the central vein. The tiles are called lobules. The lobule is the functional unit of the liver. The corners where 1, 2, 3 globules come together is called a triad because of the 3 structures it has in it. I will wait until we get to the picture, though, to talk about that. It makes a lot more sense. Function of the liver, we have lots of functions of the liver. Detoxification of the blood, maintenance of blood pressure, maintenance of homeostasis, the production of bile, as well as the storage of glucagon. The kidney slide is not going to be new to us. You saw this yesterday. Don't forget it's surrounded here, surrounding the Bowman's capsule out here, and the glomerulus with the kidney tubules, which are lined by simple cuboidal epithelium. The eye. I will not use the diagram on the left. I will show you something like on the right, and you're going to see it, and you're going to know immediately it's got to be eye, doesn't it? Well, okay, start with the dark part. That has to be the choroid coat. And then the part here where it looks like mowed grass. Maybe some of you have been lucky enough in your life not to have to ever cut a lawn. I was not so fortunate. This is the retina. And that leaves us thinking, well, there's the posterior chamber. Here's the retina. Here's the choroid coat. And this thick layer here, oh yeah, of course, that's the sclera. This tissue here that looks so spongy, okay, because of the color that it happens to be, to me it looks like popcorn floating in some sort of soft drink. But it's spongy. It has little chambers all over the place, and little bridges of cells. Very, very thin single layer. That's simple squamous epithelium. Bowman's capsule was made out of this kind of stuff, but this is lung. This is lung. And the smallest chambers that we can see, like this one and this one, are alveoli, the place for external gas exchange. Down here, don't be confused, that's probably a bronchiole. The skin, the one on the right, not going to give us any trouble. We saw it yesterday. Here's the basement membrane. Down below it is the dermis. Everything above the dermis is the epidermis. Okay, so we have 4 of the 5 layers actually in here. Impossible to say where each layer is, except for the fact that the first layer has only one set of cells, and the first layer is the stratum basale. But this last layer, the stratum corneum, has really built up quite thickly in the sample they used. We looked at skin cross-section yesterday in a nice slide. Some of the slides are not as good as others. That's the way you're going to find it on the internet. But I will definitely show you good slides of everything I'm going to ask you about. You look at this guy here, you see this, this, this. This looks like some sort of tropical fruit, doesn't it? Do you know what it is?
Like dragon fruit?
Dragon fruit is a good bet. Yes. Okay. There's something else it looks like too. Jackfruit, I think they call it, or something like that. Anyway, you see these guys. These are the oil glands. Okay. Now you know the oil gland is touching a follicle for hair, but you don't have to see that follicle in every slide. You don't see it in this slide, except possibly right here. Okay. You see different lobes here, but you don't see the attachment of this one to the follicle. And you can't expect to see that. There's fat. And then you have this one here in the center. Okay. We call it the laughing dog or the dancing dog. It's got little tubules, lots of little tubules. These can come in many different ways. I showed you one yesterday that was rounded, but this one is not. This one just happens to make a comical shape. These little tubules are the sweat gland, the sudoriferous gland. The sudoriferous gland is American gland, meaning it exudes a fluid, only a fluid. The sebum excreted by the oil gland, or sebaceous gland as it's called. Okay. This is a hologram gland because in the sebum you have entire whole cells. Here's the test, the ovary. Now this is a cat ovary because the human ovary is way too big to put on a slide. And you'd have to just take a small section of it. But here's the follicle. Here's an ovum. And the idea here is that this is a dual structure. This is part of the reproductive system as an organ, yes. It's also an endocrine gland. It produces progesterone, a hormone that counteracts estrogen. And we'll be going into that soon. But for now, realize and know that it's both an endocrine gland and a reproductive structure. The slide that gives most people trouble, so don't let it happen to you. This one is a cross-section of testes. Testes are filled with just uncountless numbers of tubules. This one down at the bottom here is nice because it shows some thickness to it, and the cells that are on the outside, where my pointer is, are the ones that begin dividing meiotically, and the two divisions of mitosis produce spermatids, which end up in the lumen. As more and more spermatids and fluid is produced, this has got to move along. So it moves towards the exit, and that's in the general area of the structure that stores spermatozoa while they mature. That structure is called the epididymis, and we'll be seeing that in a moment.
Charlie,
Yes.
are the testes endocrine glands like the ovum?
Oh, I'm sorry. That's correct. The testosterone that is produced makes the testes an endocrine gland as well as a reproductive structure. Right.
Thank you.
Thank you. Okay, and now let's move on and do our first one. Well, there's two parts. This is a handout for the organ system. Okay, it's P4 handout. It's for the practicum primarily, but please, everything that you're studying for both or all three tests. So don't try to line things up and say, I'm going to study this today and this today and this today because it's on the first test. You'll be missing a lot of questions. Got to study them all. You're going to have to organize your time better than I've been able to even, for the last 8 weeks. Both of us have to organize our time better. Okay, so I'm going to open this up. And what it is, I put it together. It's a list of things like, for instance, in the head region that you're going to be familiar with enough so that if they're pointed out, you can name them. Okay. And in many cases, or most cases, give their function. Okay. So parotid gland, that's not going to be hard, I hope. Parotid gland produces saliva for the digestion of starch and for the lubrication of food. You want to say it all, okay? But I have it in blue here, what kind of tissue it is. Primarily, it is glandular epithelium. All glands are going to be glandular epithelium. That doesn't mean that they don't have blood vessels running through them and some smooth muscle in the tunica media and things like that, but primarily glandular epithelium would be our reason for looking at the parotid gland. Okay. I haven't shown you Stenson's duct yet, or yeah, I have actually, but I'm going to show it to you again. Now I don't have any tissue type. You don't see any blue, so I don't care. I'm not worried about that. Here are the other salivary glands. We haven't done the term vestibule, frenulum. We have looked at the palatine tonsil, I believe, but I haven't told you that it's reticular tissue, reticular networking tissue that looks sort of like sponge. Okay, and that's when we get to it in these models. I'll take more time with it. I just want to get this page in front of you. Okay, epiglottis, elastic cartilage, hard and soft palate. I know you know those. Okay, I'm going to have to assume some things. And a lot of things are in the classroom notes. By in the classroom notes, actually, we should say class notes. They're in our notes that we've been using to get this far in the class. They're all on the internet. They're all in Canvas. Anytime it says in classroom notes. So you want to go back and review those if you need to. You don't need to review a lot of things. Here's the respiratory system, and here's the type of tissue that you need to look at or get it in your mind. If it says in the classroom notes, same as above. The abdominal region, we've named all these tissue types now. So now you got to sort of pair them up with the structures. So if I ask what's the lining of the stomach composed of, what tissue, you're going to say simple columnar without batting an eye. And we haven't touched the reproductive structures yet. The integumentary system, we did a few things. Okay. The fact that it says gland, sebaceous gland, actually, I should add in there glandular epithelium and glandular epithelium. So I'll look over this and do my part this weekend too. Okay, and the kidney that we were tested on, and the kidney that we lectured on. The kidney is, and the urinary system is, a substantial part of the fourth practicum. That should be a blessing. Okay, unless you have a short memory, but I don't think anybody has that, not from what I've seen. Okay, so we'll come to that. We'll come to all of that. if I had a printer, and well, of course I do, but I'd be printing out all these pages. Okay, now for today, I slipped this one in. I don't know if there's time for it. I also don't want to overload, but I definitely want to do this first one. Organs and systems. Okay. What you need to know here on the digestive system, okay? We've covered the digestive system. You've been tested on it, okay? So hopefully a lot of this is welcome material. I've explained the word merocrine to you and exocrine gland. We have three types of salivary glands, the parotid, the submandibular, and the sublingual. We can see the parotid here, and we can see the submandibular here. This model does not show the sublinguals. Exiting from the parotid gland is a duct. Okay, and this is the parotid duct, also known usually as Stenson's duct. Stenson's duct. The pharynx, we looked at it. We've seen these models before. I don't mind showing you something again, but I'll do it a little bit faster if you'll let me. Okay, here's the nasal pharynx above the uvula. We can't see it in the mirror. Not labeled here is the pharyngeal tonsil. Function of the tonsil is in the immune response, collects a lot of white blood cells, and fights off infection. The oropharynx here, the laryngeal pharynx, and the esophagus starts here. The vestibule in front of the front teeth. Yeah.
Would you mind pointing to the tonsil again?
It sounds like you're talking on a xylophone.
Oh, I'm sorry. Something in my throat, but would you mind pointing to the tonsil again? You
Okay.
said the tonsil was there.
This is the pharyngeal tonsil.
Okay.
But this one unlabeled, this number 3, you can barely see, that's the palatine tonsil. I think it's labeled further.
Thank you.
Okay, here's the esophagus. Well, in order to find the esophagus in the cat, you got to pull it from underneath the trachea. Here's the trachea, then here's the esophagus. When it says stomach, you need to know the function. So that's why it's in bold. I know you know the function is the storage of food, mechanical digestion, and chemical digestion, because we've already talked about that, and we've already had a test on that. So that's quick reading. The parts of the stomach, the fundus, the cardia, the pylorus, and the pyloric sphincter show up better on this diagram. I put it together later. And these folds are the rugae that we see inside. The lining of the stomach, simple columnar epithelium. This is the duodenum, the first part of the small intestine. It has inner folds called plicae circularis, and that is the term I will use. Okay. Helps move the contents along. Here's the gallbladder here. I took a lot of liberties with this. This little triangle or rectangle is a piece of liver. And you have the hepatic duct up here, unlabeled, the cystic duct coming from the gallbladder, and where they come together the common bile duct. We've done these before. They're headed for the ampulla vator, which is down here. And here's the pancreatic duct delivering pancreatic amylase. It's waiting to get out too, but if it can't, it will overflow and come out its own little pore right here. Here's the pancreas, part of the digestive system and also part of the endocrine system. The spleen over on this side, it does store some red blood cells. It stores white blood cells in its reticular tissue. And the spleen is also a place where red blood cells meet their demise and are broken down. This is one of those cat things that you have to look at it a while, but at least if it's labeled, it will finally make sense. Okay, here's the stomach. Here's the stomach. This looks like small intestine, a lot of it. So the head end is up here. Okay. The other reason you know this is the head end, here's the ribs. Okay. As material leaves the stomach, it first enters this tube, this part of the small intestine, and this is the duodenum. The lining of the small intestine is simple columnar again, but it has the unique presence of villi. This is a villus. Within the villi, there may be a lacteal for the absorption of lipids and fat, and also a capillary system called the lamina propria for the absorption of nutrients that have recently arrived from the small intestine. Those nutrients are not going to the circulatory system in general yet. They're going to go to the liver first.
Charlie,
Yep.
the simple columnar epithelium does not make up the villi as well?
It makes up the outer surface, yes. This is
Yes,
simple
okay.
columnar epithelium.
Okay. Thank you.
The large intestine. The models are always nice because they're so clean, you know. But when the ileum, the last portion of the small intestine, empties into the large intestine, it empties into the cecum. This is also known as the colon, of course. The cecum has attached to it, unless it's been removed, the appendix, which is right here. Function of the colon, again, is reabsorption of water, compaction of waste. Ascending colon, hepatic flexure, transverse colon, splenic flexure, descending colon, and when you enter into the true pelvic cavity, this is the sigmoid colon. These light areas, the bands that you see, you don't get to see them all at one time. There are 3 bands. Those are the tinea coli, and their irregular contraction causes the formation of hostra. The singular is hostrum. Here's one over here. There's so many of these hostra. The hostra help to move the contents forward. Another photo, and this one again, you'd have to take a while to think it through. I think if I were to give you this one now, almost everybody would miss everything. But if I were to show it to you right now, and we come back to it one more time, and I put it on the exam, I think then you'd probably appreciate it. Well, this is the small intestine. That's the first thing we can say. Let's follow it along. Okay. and it comes in here and it enters into this body. This is the ileum. There's a valve right here between the small intestine and the ileum, and this large structure, which is the colon. This is the ileocecal valve right in here. Tough one. And this over here is the cecum. Sort of curves down a little bit. And then the large intestine is here. This would really be the ascending colon.
Charlie, can you point out the stomach in that picture, please?
It's not there.
Ah, okay.
Okay. I don't see it either. I will not show you a slide on the practicum that I don't feel you've had enough time to look at and understand. You haven't had enough time on this one, and I think I can find a better one anyway. Okay. The liver. Okay. But this slide isn't bad at all. Here we can see the diaphragm. We can see some pieces here of lung that aren't labeled. Here's the heart. Here's the liver. And here's the gallbladder. This is spleen over here. And this mess is actually in good shape. That's the way it should look. This is the greater omentum. It's attached to the stomach. Well, where's the stomach? Fortunately for us, it's underneath here. But this is the greater omentum. I believe I showed you this before. Is that correct?
I don't believe so.
Okay.
I don't think so.
Okay. It's quite possible. I mean, I just finished another class, and then you guys came along, and so things sometimes run together. Okay, this is a daunting picture. Like, how the hell do you make anything of it? But you'll have no trouble with it, really. Let's do it together. This is the liver. Okay. Now, we're actually looking at the liver here from the top, and the slice has been made so to take the top off. Then if you know that's the liver, this structure here is almost universally green. This is the gallbladder. Okay. Where did I say just a moment ago, blood will arrive to the liver from? I don't know if that made sense. After blood is picked up by the small intestine, it does not go into general circulation. It comes here for the liver to detoxify it. Okay. And here's where it comes in. This blood, okay, has new nutrients picked up in the small intestine, and they're not going to get a chance to run throughout the body and possibly poison the body. So the liver is there to detoxify. The blood comes in from the small intestine from this vessel, which is the hepatic portal vein. Okay. When the blood that is suspect, let's call it, gets into the liver, it spreads out. You can just follow the color everywhere. And it's divvied up into those lobules that we saw a little bit earlier. filters through the lobule from outside to inside, from outside to the inside central vein that we looked at. And in so going past all these cells, those cells detoxify it. Is one pass going to be enough? Not necessarily. If you're not sober, one pass certainly isn't going to be enough to get you past the speed trap or a sobriety checkpoint. You've got to have quite a few passes of the blood through the liver in order to detoxify it. But the first time through is important. It does the best job. And all the blood that's suspect goes into the lobules. Now, it goes to the lobules and to the central veins, and they all interconnect. The blood's been looked at, been examined. It comes back through these blue vessels. All of these blue vessels bring the blood back and dump it right in here in the inferior vena cava, which is on its way back to the heart. The blood also needs to come into the liver, not in an impure or suspect state. The liver needs good blood for itself, like any other organ does. And the hepatic artery where I am right now, brings good blood from the systemic circulation. Good blood flows out red to all parts of the liver, just like the suspect blood does. So you've got good blood flowing through, and let's call it bad blood flowing through. Okay. Once the bad blood has been processed and the good blood has been deoxygenated or used up, both of them exit into the vena cava. The falciform ligament that supports the liver is shown here. We'll probably see this at least one more time, and pretty soon it'll be your favorite. Okay, here's the liver. Start here. Here's a lobule. Here's a central vein going down a lobule. Another portion of a lobule with a central vein. And here, where 3, 1, 2, 3 of these come together, where 3 of the lobules come together. This space is a triad. This space is a triad. It's called a triad because when we look at it, here it is. Part of it has the portal vein coming from the small intestine, dropping the blood into this labyrinth of passageways, being detoxified by the cells surrounding it until the blood reaches the central vein. Also, here's the hepatic artery, a little one. This is bringing good blood to the liver. When we use the terms bad blood and good blood, they're right together. Here's the bad blood, here's the good blood. And the other part of the triad, the third part, is a bile duct from the from other parts of the liver or from this part of the liver. It's headed on its way downward to the common bile duct. This little,
Charlie,
yeah.
so the term triad refers not only to the 3 lobules of the liver coming together, but also to the bile duct, hepatic artery, and portal vein that meet in that space?
Yes, it does.
Okay, thank you.
Okay. The liver, largest gland of the body. All glands are composed of glandular epithelium with one exception, and that's the neurohypophysis. It's misspelled. There's not two P's. Sorry about that. Neurohypophysis is the posterior part of your pituitary gland, and it's nervous tissue. We'll be talking about that soon when we talk about endocrine glands. Other than the neurohypophysis or posterior pituitary, all glands are composed of glandular epithelium. I think that's a cop-out, really. You can't tell me that liver cells are the same as salivary gland cells. You know, there's got to be a difference, and but they never put it into the textbooks. It's glandular epithelium, so we'll leave it that way. Here's a summary of important tissue types of the digestive system and the type of epithelium or cell that composes them. Okay, before I let you go, well, you're always free to go. At the bottom of today's material, okay, are the cranial nerves. Okay, and you'll be asked to, in the lecture test coming up on Wednesday, you'll be given Roman numerals, a few, not a ton of them, but 3 or 4 or 5 at the most. You'll be given Roman numerals and asked to name the cranial nerve. Okay. And that's it. I'll just tell you right now, that's it. Don't worry about the sensory or motor function for this test, for anything that we have yet to do. Just these 2 columns. Okay. And then the bonus exercise I emailed to everyone. This is due a week from tomorrow. Okay, it just asks some general questions, some very easy. Name the 4 tissues types. Then it asks you to define a few words. Okay, and what tissues make up these organs, and so on. It's not all on histology. They have questions on hormones that we haven't yet gone over. I imagine I'll be there on Monday. I have to be on Monday and Tuesday because that'll be our last lecture time. We won't lecture on Wednesday. And although on Wednesday I will have a Zoom meeting to discuss Thursday's exams with you. Okay. This you have either here or you have it in your mail. If you're the type of person that likes to go ahead, please don't go ahead to the spinal nerves lecture. Please don't. Okay. Okay. Now, if you have any questions, you can hang around, or you can ask a general question right now if you think it would be the good of more than just yourself. Okay, then. I'll see you tomorrow.
Thank you, Charlie.
Thank you, Charlie. Have a good day.
Thank you, Charlie.
Bye-bye.
Thank you, Charlie. Have a good night.
Bye-bye.
Here I go again. Thanks, Charlie.
Okay.
All right, see you tomorrow. Bye-bye.
Bye-bye.